Chronic inflammation is kind of integral to MDS. So it's been known for a while that chronic inflammation, low-grade inflammation, sterile chronic inflammation happens in myelodysplastic syndrome. It's been known for quite a while, but only in the past few years, it's kind of come to attention enough that there are now therapeutic approaches that are in development targeting these. In fact, chronic inflammation is not only stigmata of MDS, but it's also the same for CHIP or clonal hematopoiesis, and also for aging. So it's all like kind of interrelated. They call it the inflammaging, and I think the VEXAS syndrome, that's a recently discovered syndrome that links the autoimmunity with the myelodysplastic syndrome and the UBA1 mutation. It's kind of a prototypic example of how integral chronic inflammation is. So the biology of MDS, it's multifactorial. So you have the hematopoietic stem cells and progenitor cells. These are abnormal because of the mutation, right? Clonal cells. And then you have the bone marrow microenvironment that has the macrophages, T cells, or important components of inflammation, the myeloid-derived suppressor cells that play an important role in supporting these abnormal hematopoietic cell survival. And then, of course, the pro-inflammatory environment. It's multiple evidence out there showing that the innate immune system, the toll-like receptors are activated in MDS, and that kind of triggers the downstream after CD33 binding, the IRAX, further down the NLRP3 inflammasome, that kind of links all these the mutations with the bone marrow environment and the inflammation all together, resulting in a phenotype of myelodysplastic syndrome leading to cytopenias. And I think the inflammation I think this kind of integrated approach is the one that kind of explains why patients with different mutations, different mechanisms, all boil down to the same phenotype of cytopenias that's typical for MDS.